In this series of lectures that we are going to have today, first thing I'm going to talk about briefly the virus that you are hearing a lot about these days. Uh, Coronaviruses are not new viruses. I'm going to discuss coronaviruses briefly. It is a large family of viruses which are basically single positive stranded RNA viruses. And the word corona comes from the it is a Latin word coronum, which means that these viruses have a crown-like appearance, as you can see in the figure there, and they have spike glycoproteins on their envelope. Now, the subfamily is Orthocoronavirinae, which classifies into 4 genera of coronaviruses: alpha coronaviruses, beta coronaviruses, delta, and gamma coronaviruses. The beta coronaviruses are the one which we are dealing with. This COVID disease is caused by a virus known as SARS-CoV-2, which belongs to this family, beta coronaviruses, which further divides into 5 subgenera. Now, the genomic categorization is important to know. Clinically, that it has been shown that bats and rodents are the genetic sources for alpha and beta group, while avian species seem to represent the genetic sources for delta and gamma group. What is important and interesting about these viruses, the coronavirus in general, is that it can cause cross-species transmission, and therefore from animals to human transmission is noted in these viruses. Now, in humans, the illnesses can range from minor common cold to severe diseases. And the severe diseases is basically, they are the major pathogens in the recently diagnosed respiratory disease outbreaks, which you already know. There was SARS in 2002, which was, that time it was very serious public health concern itself. And this property of theirs to cross interspecies makes them potential risk for a pandemic, as we are going through one, and it is therefore a serious public health problem. Now, till now, in humans, only 7 times these coronaviruses have been isolated as a cause of disease. Earliest was in 1960s, where you had 2 written species. I'm not going to name them because there's no point of discussing them. But recently, there are SARS in 2002, another one in 2004, then 2005, and then again we have heard about MERS, which was again from the family of coronaviruses in 2012, and now in 2019 we are dealing with SARS-CoV-2, and most of them have caused serious respiratory tract infections. Now, it is not like that coronaviruses don't exist in human population. So in general, 2% of our population will be healthy carriers of coronaviruses. And these are responsible for causing 5 to 10% of acute respiratory tract infections, which could be upper respiratory as well as lower respiratory. Now, this will depend on a person's immunity, also on age and genetic and ethnic factors. So, common human viruses will cause common colds and self-limiting URTIs in immunocompetent people, and in immunocompromised host and elderly, LRTIs can also happen. Recently, these SARS disease in 2002, the COVID disease, and MERS, they have caused epidemics of high clinical severity with both respiratory and extra-respiratory manifestations. What is concerning is the mortality rate. That is why we're all worried with this disease also. Earlier, SARS had a mortality rate of around 10%, and MERS had a mortality rate of around 35%. The current mortality rate for COVID-19 is somewhere around 10 to 15%. Now, SARS-CoV-2, this is the virus that we are going to talk about. It was initially known as novel coronavirus 2019. But later, since it was found to be very similar to the SARS virus, which caused the outbreak in 2002, the Virology Society internationally decided to name it as SARS-CoV-2 virus. As I told you, it is from the beta coronavirus category. It is pleomorphic in shape. Interestingly, it is sensitive to ultraviolet rays, and it is also known to be heat sensitive. It can be effectively inactivated by lipid solvents like ether, ethanol, chlorine-containing disinfectants, peroxyacetic acid, and chloroform, but not chlorhexidine. Now, the dynamics, how it came to humans, for this virus, it is not clearly known how it came to us, but the speculation, again, it has an animal origin. Genomic analysis suggests its evolution from a strain found in bats, and potential amplifying mammalian host. It is still not decided which one it was, but again, it has to be a mammalian host. There was some talk about pangolins being one, but it is still under speculation. Another theory suggests that the original SARS strain, which happened in 2002, it got mutated, and now it could have directly triggered virulence towards human, but again, this would point that there is no intermediary mammalian host. Coming to the transmission. So first cases, as it was reported, all of us know the city now, Wuhan city, it was linked to the direct exposure of Hunan seafood wholesale market, where animal to human transmission was presumed to be the main source of infection. Subsequent cases could not be explained because of this mechanism. So animal to human was the first mechanism speculated, but then since it was so rapidly spreading, it was decided and it was concluded that it had human to human transmission. Although symptomatic people are most frequent source of spread, but the possible transmission in asymptomatics through asymptomatic people cannot be excluded till date. Moreover, there are suggestions that individuals who remain asymptomatic could also transmit the virus. So there is at this point of time, there is enough data to suggest that use of isolation is the best way to contain this epidemic. Now, further moving on for transmission, the data from China again seems to indicate that close contact between individuals is necessary. So transmission, how is it happening? There are two main routes. First is the droplet infection. So respiratory droplets which get produced from someone coughing, sneezing, or loudly talking. So as we can know, sometimes we can visibly see them. Droplet infection directly makes a contact with the person, and that person, the contact can be in the eyes, in the nose, in the mouth, and then in the hands, and then we are touching our faces, so that can infect the person. Another route which has recently been found to be there, and it is again under study, is aerosol transmission, which is also a possible cause in cases of exposure when the aerosol production is in the closed spaces. The incubation time is somewhere between 3 to 7 days and up to 2 weeks, as the longest time from infection to symptom which was studied was 12.5 days. And on an average, each patient transmits the infection to additional 2.2 individuals. Someone had asked me earlier also, many of us really don't know what is droplet contact and what is airborne transmission. So infected droplets contacting surfaces of eye, nose, or mouth generated when an infected person coughs, sneezes, or talks. Those produce infected droplets. These also cause short-term environmental contamination like a soiled surface, doorknob, handrail, anything which the droplet fell on, which the other person can pick up in a very short span of time. Now, droplet aerosols are also produced, which are final mist, which are generated during procedures that we are exposed to, particularly the healthcare professionals. So it is intubation, bronchoscopy, suctioning. So that is why we are more at risk than the general population. Droplets are too large to be airborne. So airborne is something which is suspended in the air for a longer time. So droplets will settle down and they'll go out of air. Droplet transmission is reduced with the use of hand wash, sanitization of your hands, cough etiquette, using protective personal equipment like face masks and goggles. And the diseases which are spread by this method are measles and SARS. So SARS, again, both previous one and the current one. The airborne infection is situations where the droplets settle down, but their residues evaporate to finer droplets, and the dust particles again carry these infection microorganisms, and they remain suspended in the air for longer periods. Now, for this to happen, the organism should be such that it should be able to survive for longer periods outside a host and must be resistant to drying. And then it may enter your airways. So this theory is still being speculated for SARS. It may not be there, but then they have not still ruled it out. The most common airborne disease that we all are dealing in our country is tuberculosis, and we also know that flu-like illnesses spread through droplet infection. So going by the route of transmission, now we know that who's at the risk of infection. From a person, it's not just the persons who are in close contact, it is actually everyone in the community. And last slide, I will tell you about what is this big thing about this contagiousness about this virus. So first of all, we have understood now that there is insufficient rapidly evolving theories regarding the potential routes of transmission. So droplet is confirmed, airborne is still speculated, and there is some data that the virus gets secreted in the body secretions like stools. Symptomatic patients, though, are definite shedders, but estimates will be refined as more data becomes available, and more information is needed whether transmission can be done from asymptomatic individuals or even during the incubation period. And lastly, researchers have also found that this virus, the spike on this virus is having an avidity of 10 to 20 times to bind to human cells as compared to the previous SARS. So now it is spreading more easily person to person than the previous SARS even. Again, isolation is the best way to contain. Thank you.